Muscles are made up of sarcomeres kept in series. So if you look at each sarcomere, you have thin filaments and thick filaments. The green are the thick filaments, and in red you have the thin filaments. The thick filaments are made up of myosin molecules. Myosin molecules have a head, a neck region, and a tail. The tails are coiled together. The thin filaments are made of three molecules. Actin molecule, that's in blue. Then you have the green tropomyosin molecule, that lies between these strands. And then you have the troponin molecule. The core concept of skeletal muscle contraction is called the sliding filament theory. The thin filaments slide over the thick filaments. Due to the sliding of the filaments, the Z lines have come together. That is, the sarcomere has shortened in length. The basis of sliding filament theory is the cross bridge cycling or the walkover mechanism. In orange, we have the myosin filaments, that is the thick filaments, and you can see the head of the myosin with the neck in orange. In maroon, we have the actin molecule, which is a long strand, and you can see orange active sites on the active molecule. You can see the blue tropomyosin molecule has covered the active sites of actin, preventing the interaction between the myosin head and the active sites. You can also see the green troponin molecule in this picture. Troponin has three parts, I, C and T. Troponin C binds to calcium. When calcium comes and binds to troponin C, it undergoes a conformational change. The shape of troponin changes. When troponin changes, the tropomyosin which is connected to the troponin moves, and now the active sites of actin are exposed. The myosin head can now bind. Once the myosin head binds, it undergoes a power stroke. It will go from 90 degree to 45 degree. An ATP binds to the myosin head. Using the energy of the ATP, the myosin head goes back to the 90 degree position. That position is also called the cocked position, the way the gun is cocked. Now the myosin head can bind to the next active site, and that again will undergo another power stroke. That keeps on pulling the thin filament towards the center of the myosin. When the contraction has to end, the calcium is pumped back into the sarcoplasmic reticulum by Cerca. When calcium concentration decreases, the troponin will go back to its original state, and tropomyosin will now come and cover the active sites of actin.